At the most recent EAHAD meeting, Dr. Johnny Malangu presented on behalf of GENEr8-1 investigators the 2-year follow-up data from the BMN 270 phase 3 study. So this study is evaluating the safety and efficacy of Valoctocogene Roxaparvovec, or Valrox. And this consists of an AAV5 vector with a factor VIII transgene that targets the liver. This was administered as a single infusion of 6 × 10 to the 13th vector genomes per kilogram, and in an intent to treat population that included 134 men with severe hemophilia. Now, of those, 112 were enrolled from a non-interventional lead-in study where they were followed for at least 6 months on standard of care factor VIII prophylaxis. And this allowed us to establish their baseline factor VIII utilization and annualized bleed rates. Now, following the gene therapy infusion, prophylaxis was then discontinued, and we had an opportunity to evaluate the efficacy based on endogenous factor VIII levels that were achieved, the annualized bleed rate, and the need for factor VIII infusions that was ongoing. So the mean factor VIII activity achieved by chromogenic assay was 23 international units per deciliter at year 2 post-gene therapy treatment. As has been seen in all other gene therapy trials to date, the factor VIII activity as measured by the traditional one-stage clotting assay was actually about 1.6-fold higher at 36 IUs per deciliter, so just shy of the non-hemophilic range. The impact of achieving these factor VIII levels is that mean annualized bleed rate decreased from 4.8 during the lead-in on factor VIII prophylaxis to less than 1 post-gene therapy, so an 85% reduction. And accordingly, the mean annual number of infusions decreased by 133 compared to the lead-in phase, so a 98% reduction. And this was impactful for patients as reported by the HemoQual-A quality of life instrument, which showed a statistically and clinically meaningful total score reduction. There were no new safety events and no new treatment-related SAEs that were observed during year 2. Overall in this study, 83% of the participants have experienced liver enzyme elevations that triggered an immunosuppression regimen, most commonly oral corticosteroids, but 99% of the patients are off immunosuppression by year 2. Now, some important caveats of these observations from the trial so far is that we have seen considerable variation of more than 10-fold for the range of factor VIII levels that have been achieved amongst the subjects. And in addition, the mean factor VIII levels for the entire group have declined from the levels that were seen at the end of the first year by as much as 40 to 50% in some individuals. Nevertheless, these factor VIII declines were not accompanied by a reduction in control of bleeding in the vast majority of the subjects. So bottom line is that a single treatment with Valrox was well tolerated. This has led to stable and durable bleed control that's superior to standard of care factor VIII prophylaxis through 2 years. So we're going to look forward to continued follow-up of these patients in the coming years to assess the ultimate durability of this intervention.